Okay, I think everybody is back. So obviously we had some issues with the Genome Browser. That happens sometimes when there are multiple people accessing the same region in the Genome Browser. But obviously in normal circumstances, you won't have 100 or whatever, 60 people accessing the same location at the same time. So hopefully that's not a standard issue. As usual, if you're ever using the Genome Browser and you start seeing things like you saw today, definitely let us know, because that helps us troubleshoot and figure out what network connectivity issues are causing these problems. So what we're going to do, or what I'll do right now, is we're going to talk a little bit about exploring gene models in the Genome Browser, and then we'll take a break, and then after the break, we will go straight into an exercise where you'll get to, in the group, you'll get to explore some of the gene models and make your own conclusions about them. So, one thing to note, this day and age with, especially with most of the genomes having various types of RNA sequence data available, you can actually make your own conclusions about the gene models. Many of the gene models, if you're lucky, the genome maybe had some manual annotation, but in most cases, many of the genomes are automatically annotated with the gene models. And depending on when they were annotated, the gene models may be less accurate if they were, for example, completely based on some older algorithm, or maybe some blast similarity when there weren't many gene models out there. And so having RNA sequence data available is quite useful because it's real data, and you as a scientist can look at the data and make your own conclusions about the results. So let me see if I can try and do this. Let me stop sharing again. And okay, I'm assuming everybody can see the whiteboard, hopefully.
Yes.
Okay, so typically when you, I'm just going to draw this. So let's say this is a line. This is going to be very, very crude. This is your genome. Okay. Sorry, my typing on this will be terrible, but hopefully you'll get the point. So here's the genome, and you can imagine that you have a gene here. So with 2 exons. Okay. So let's do another one. Okay, so okay, so this is exon 1, and this is do another one here, exon 2. Okay, all right. So exon 1, exon 2. Okay, so so when this gets transcribed into RNA, what's going to happen is your exon is going to be transcribed into something that will ultimately be spliced together. Your genome, this particular gene will be transcribed into something that looks like this. Okay, so we have exon 1 is here and exon 2 is here. Okay, and then what we do is when you do an RNA sequence experiment, you're actually sequencing this, right? So we send this to sequencing, right? And what's going to happen now in your sequencing with next-gen sequencing, you're going to get a bunch of reads that are coming from this particular transcript, okay? So you'll get millions of reads, or hundreds of reads, or thousands of reads from this transcript. So then the next step, typically when you have these reads, what you do then is you take them back and align them back to the genome. Okay, so this comes back here, and you align to genome. Okay, so now what happens when you try and align these reads? So some of these reads, and we can change the color here. So some of the reads are going to align to exon 1, right? Because the expression came from this region where exon 1 is present. Some of the reads are going to align to exon 2 because they're going to be coming from the region where exon 2 was expressed. Okay. But then some of the reads are going to align, they're going to try and align to the junction of where exon 1 and exon 2 came together, right? So the reads that come from here, if you try and align them back to the genome, there's really no place for them to align unless you have an aligner that is what's called splice aware, right? And many aligners are splice aware. And so what happens is when it's splice aware, these reads that are coming from this region are going to align partially here, and then they're going to be split, and they're going to align to this region here. Okay, so hopefully that makes sense to everybody. So now these reads that are split, right? Anytime a read gets split on the genome, because a part of it aligns to one exon and the other part aligns to the second exon, this is evidence that this intron exists, right? So this is actual real evidence, real RNA sequence evidence that this intron exists. And the more reads you get that align to this region, the more evidence you have that this intron exists. Okay. So we take advantage of this. All right. So I'm gonna stop the whiteboard sharing. Hopefully that wasn't too terrible, but hopefully you got the point that that we can take now this information that we get from the reads that are coming out of the transcripts, and we can align them back to the genome, and we can use that information for different things. So the reads that are aligned to the exons tell you something about the expression of the exons. The reads that align to the junction will tell you something about the evidence as to whether the intron exists. And so, of course, you can imagine that this gene model is incorrect. So let's say this gene model is incorrect, and in fact, there's another exon right here. Let's call it, this is exon 0, that really belongs to this gene, but wasn't part of the annotation. And so what's going to happen is when you do your sequencing, you would have gotten lots of reads that came from this region here, right? This fictitious, let's add this exon here. So this hypothetical exon is will be spliced right here. You'll get reads from here, and then when you align them back to the genome, they're going to align to this region where in the genome or in the annotation there's no exon, right? So you're going to see a pileup of reads. It'll look, imagine there's another gene right here that wasn't annotated. There are going to be a bunch of reads that are going to align right here, and then you might look at this and say, wow, this looks like next to my gene there's another potential gene expressed, and on this side of the gene it looks like there's potentially another exon that belongs to my gene, because I see there are reads in this region, and there are also reads that are split between my exon 1 and this upstream hypothetical exon, right? Okay, so now from this RNA sequence data, we can get evidence of the expression of the gene, we can get evidence for splice junctions, we can get evidence for new genes, and we can get evidence for new potential alternative splice versions of a gene or exons. You can imagine here, let's make another hypothetical scenario. Let's do a different color. This will be green. Imagine now you had a read that had to be split between this exon and right here, this exon right here, okay? So you can imagine now having what's called an exon skip polymorphism. So you can have an alternative gene model where this exon right here is spliced with exon 2 and it skips exon 1, okay? And so now when you're looking at this, you will say, wow, well, this gene is not only has expressed potentially as a 2 exon gene, it may have a third exon that's upstream, which sometimes all 3 of them are spliced together, but there are some cases, maybe it's a developmental cycle, or maybe it's just an alternative splice variant, that where the exon 0, let's call it, is spliced to exon number 2. And so now you can generate a new gene model that's an alternative gene model for this particular gene. Okay. All right. So are there any questions about the splice variation, or sorry, the utility of RNA sequence data for identifying intron splice junctions or new genes. Okay, any other comments from the other instructors? Okay, I'm going to close the whiteboard then. All right, so let's go back and go back to our databases. And so we take advantage of this RNA sequence data in different ways. So I'm just going to go to, we can pick ToxoDB, go back to the homepage. And so there are a couple of things that we can do, or that we do with the RNA sequence data. So one, hopefully you saw during the exercise right now, is that we can align the reads and look at them in the Genome Browser. And we can look at the coverage plots, and I'll show you these in a second, or we can look at the splice junctions themselves that I was just showing you on the whiteboard, and we can count how many reads support a particular splice junction. And in fact, what we did recently, this is actually not part of the exercise, but I will show it to you here, and hopefully each of the instructors in the breakout rooms, if this is available, actually it is available, ToxoDB, make sure you demo this as well, or make sure everybody in the breakout rooms is aware of this. But if you go to our searches and you just type intron, you'll find that we have under the gene model section, we have an option to look for unannotated intron junctions. And if you select this option, you can select your organism of choice. So let's say I'm going to pick Toxoplasma gondii ME49, which is the reference strain. And I can go in here and I want to find any gene that has unannotated introns. And I can now specify how many reads support this particular unannotated intron, and also what percentage they are of the most abundant intron for that particular gene. And so, for example, I can say, well, I really want something that's really has strong support. So I want to pick something that has at least, I'm just going to randomly pick 500 reads. The answer may be 0, as always, as I've mentioned to you yesterday, you can get an answer of 0. And I'm going to say, I want it to be between 60 to 100% of the most highly abundant intron for the gene that's returned, okay? And so then you can run this search. And so just as yesterday, this will start a search strategy and return the genes that met this criteria, if any. And it turns out there are 796 genes that have unannotated introns that are between 60 and 100% of the most highly abundant intron for that particular gene. So that's interesting. And so we can take a look at those. So I'm going to go and pick, I don't know, let's pick this NTPase2 gene, and I'm now looking at the gene page. And if we scroll down and look at the gene model section, this will open up the Genome Browser view, which shows you the gene model right here. And so it's highlighted, and you can see that there's it's a 2 exon gene. And then as you scroll down here, this is all the evidence, and there are 2 subtracks as part of the intron junction support. So these are coming from these split reads, right? So we're counting all these split reads, and we're returning the number of split reads that support this intron. And so I'm going to go ahead and open this in the Genome Browser in a separate page and let it load behind the scenes. But you'll notice here there are 2 tracks that are subtracks that are open automatically under the evidence for introns. One is called matches annotation. Okay, so these are introns supported by the reads that match the annotation. So if you look at the annotation right here, it's a 2 exon gene, and if there's an intron that spans this exon and that exon right here, if there are reads that are split right here, those will be reads that support the actual official annotation, all right? And then you have a section called unannotated with strong evidence. Those would be the reads that I looked for. And in this case, you'll notice that they're actually, none of the reads supported the official annotation, but there are reads here that support there are reads here that have strong support, but are not necessarily part of the official annotation. Okay, and so then that's something that's worth looking into a bit more closely, especially if this is a gene that you are working on. And so let's take a look at this. Hopefully it opened up. Yeah. So now I'll open it up in the Genome Browser where you can interact with this a little bit more. So there are a couple of things you can do. I'm going to go ahead and pin my, my transcripts to the top. That's nice because now I can add additional tracks and I can move around and my gene models stay at the top here. And now we can take a look at this and you will see that there is an intron that goes from all the way up here, okay, and comes all the way down to the middle of this gene. And there are additional introns right here. And at some point, you start realizing that, wow, there's a bewildering number of possible splice variants or splicing going on in this region, right? And so if you were somebody who's curating this gene, what would you do? How many gene models would you call based on this region? So you can see that it gets super complicated after a while, right? But we're going to try and do this. So during the exercise today, we'll try and explore gene models and look at them and determine if we can come up with some conclusions that we can use to potentially make a change in the gene models. So here I only showed you the track for intron splice junctions, but there are many more tracks that you can turn on that could be useful for you. And so, for example, I can turn on some ChIP-seq tracks. And so if we look here on the left-hand side, you'll see there's ChIP-seq data. And then looking here, you can start taking a look at these, but they are, so ChIP-seq is basically a chromatin immunoprecipitation followed by sequencing. And basically what you're doing is you're cross-linking different histones or DNA binding proteins. So there's you're cross-linking to the DNA. So you're basically freezing the status of the epigenome, basically, and then using an antibody specific to the protein that you're interested in. Let's say a modified histone. You can you chop up the DNA that's been cross-linked, and then you immunoprecipitate the antibody, which is cross-linked to immunoprecipitate the protein, which is cross-linked to the DNA. And then after you do that, you remove the cross-linking and you sequence whatever's left over. And whatever comes back in your sequence is where your modified histone was binding. So now you can sort of look at where the genome is marked based on these different modified histones. And if you go down here, you will see that there are different types of histone modifications that you can use to, and I only want to look at the unique ones, that's good, that you can use to look at potentially the location of promoters. Of course, there's a lot of information about these different histone modifications. I would always recommend that you go back to the paper where this came from and see, well, how were they describing using these modified histones for their analysis. And I'm just turning a few of these on that I think should be useful in determining the expression here. I don't want to look at the combined track for now. Okay, so you can see here. So let's say you don't know anything about these, about what these histones do. If I look at this histone, H3K K4 methyl 3. So this is a methylated on the lysine number 4, and it's histone 3. And so without knowing anything about what this histone is doing, you can take a look and see that, well, it's actually wherever I'm looking here, there's a beginning of a gene, and I see there's a peak. Here's potentially another beginning of this annotated gene, and I see a peak. Here's potentially another one. There's some smaller peaks here. If we move down a little bit, does this align? There's a peak right here. Maybe this is a double peak, right? And so I think, without even knowing what this particular histone mark here, the middle one does, you can potentially conclude that, oh, maybe this is marking the beginning of the genes, right? And I think this is accurate for this particular histone. So looking back at this region, we can take a look and say, well, is there a transcription or potentially a promoter region upstream of this gene? And you can see that, yes, there is one right here. There may be another one right here. And so the question is, do any of these match the intron splice junctions? I don't think they do at this junction here. So this is interesting, but you'll also notice that there are these other potential tRNA genes right here. And so maybe these are marking where the tRNA genes are. And so it gets very complicated, but using this data in combination, I probably picked one of the more complicated genes to look at to try and annotate, but using this different information, and we'll do that in the breakout rooms, you'll be able to then come up with some conclusions about the potential annotation of a particular gene. Okay, one thing I wanted to point out, when you look at the annotation of a gene, you'll notice here there's a community annotation from Apollo track. And this is annotation that you can contribute as a scientist and somebody, you know, if you work on toxoplasma and you take a look at a gene and you realize, wow, this gene model is incorrect, and you have some evidence either from your data or looking at the RNA sequence data that's very strong that tells you that this gene model is, there's potentially an alternative gene model, you can actually use a tool called Apollo to generate your own gene model and submit it. And then the next day, this will appear right here, and anybody in the world can take a look at your contributed annotation. This is really, really nice, you know, and a very nice way for everybody to contribute their knowledge and their information about genes. And later today, we will have a short discussion about this, and Uli, who's one of our expert annotators, will be leading that discussion. Although we have other expert annotators on the call as well. So everybody will be able to help with showing you how Apollo can be used for making changes in the annotation. Okay, so this is all I was going to say about this. So when we come back from the break, I think we have half an hour. So in half an hour, we'll come back and we'll go into straight into this exercise again in groups. But hopefully we'll have a little bit more active discussion in the group. And the idea is that each group, you're going to configure your Genome Browser track to look something like this. You know, I would recommend that you first try and do it yourself, but there is a link here that will take you to, I think it looks similar, maybe not 100% this, but it'll look similar enough. And so the idea is that you take a look at a view like this in the Genome Browser, and each group, so depending on how many groups we have, but based on your room number, your breakout room number, each group will take a look at 3 genes, explore them, and come up in a short time, come up with some conclusions as you think that if this gene model is good, bad, would you change it? How would you change it? If we have time when we come out of the breakout rooms, we have about just a short time to discuss. We might discuss 1 or 2 of the genes from the groups. If there was anything super interesting or people had questions about, we'll put it up on the shared screen and discuss them together. Okay, with that, are there any questions before we go on break? Okay, so Rachel, we can go ahead and go on break. Everybody, we'll come back in half an hour.